There are many ways for things to go across the cell membrane. One of them is diffusion, which is the net movement of particles along the concentration gradient. So this type of movement goes from high concentration to low concentration region. It is useful for cells to obtain useful materials such as oxygen and nutrients like glucose and amino acids. This is also a way for cells to get rid of what they don't want, such as carbon dioxide, and it's needed to distribute materials in the cytoplasm. To get a higher diffusion rate, you would want the concentration gradient to be steeper, the temperature to be higher, the particle size to be smaller, the diffusion distance to be shorter, and also a larger surface area. You can imagine this in the scenario of gas exchange, where the steep concentration gradient is maintained by having the blood vessels to bring away oxygen when it's absorbed into the blood. To achieve a shorter diffusion distance, your air sacs only have a 1 cell thick epithelium, and the wall of the blood capillary is also only 1 cell thick. And finally, you achieve a large surface area by having so many air sacs in the lungs. The next method is osmosis. So osmosis refers to the net movement of water, only for water, across a differentially permeable membrane. A differentially permeable membrane is one that only allows certain things to go through and some things cannot go through. So from a region of high water potential to a region of low water potential. This is osmosis. Now let's say you have a cell that is an animal cell and it is inside a high water potential solution, one that has a higher water potential than the cell itself. Then water will move into the cell continuously. So water will keep going into the cell until when? Now the cell, because it's an animal cell, it doesn't have a cell wall. So when water goes in continuously, the cell will first swell. The cell will get bigger and bigger and bigger until when the cell cannot withstand this pressure anymore, it is going to burst. Now this time, let's imagine the animal cell in a solution with a lower water potential, or a hypertonic solution. In the hypertonic solution, the animal cell is going to keep losing water to the solution because water moves from a higher water potential area to a lower water potential area. The cell will then shrink because it's losing its water, it's losing its volume, and eventually it would get wrinkled. Now let's imagine the scenario in plant cells. So the plant cell is put into a hypotonic solution. By the way, a hypotonic solution is one that has higher water potential but lower solute potential. So the plant cell is now in such a situation. Because of the high water potential outside the cell, there will be a net movement of water into the cell, from outside into the cell. So the cell will be absorbing a lot of water in this case. The cell will then become turgid. It will not burst because the cell wall is strong enough to support the cell. In this case, the cell will be hard and strong enough to stack on top of each other and support the plant. Now this time, let's think of the cell in a hypertonic solution, one that has lower water potential but higher solute concentration. Because of this, water will move out of the cell. There will be a net movement of water from the cell to the environment. Because the cell will be losing a lot of water, it's going to lose its turgidity. The cell is going to become flaccid. Flaccid is basically a fancy word for becoming soft. So in this case, the plant cells will not be hard and strong enough to support the plant. In some cases, there will also be plasmolysis, meaning that the cell membrane and the cell wall, they separate from each other. The vacuole will also be smaller because it is losing a lot of water.